Today we will discuss about the resident skin bacterial species changes at using these phylum labels. In the previous video we have understood that if the mice undergoes this cis UCA treatment or UVP exposure, then it will cause change in the resident skin bacterial species. Here they have used this phylum label or 16S RNA data to find out the changes in the residence skin bacterial species at different time intervals after the treatment with this cis, either with this cis UCA or exposure to this UVP. So first we will discuss the results of this phylum label at 8 hours and compare it with the baseline. In the baseline they are able to see that most of this resident bacteria belongs to this proteobacteria, and other bacterial species is almost uh, equally divided into the four different species, that is actinobacteria, bacteroidetes, firmites, firmicutes, and pastishubacteria. Pastishubacteria. However, after 8 hours, when they analyze this 16S RNA data, they are able to see that there is an increase in this firmicutes and there is a relative decrease in the actinobacteria and proteobacteria. So this is for the mice that are treated with this cis UCA. The mice that are undergone with this UVP exposure will also has also shown an increase in firmicutes, and there is a relative decrease in the proteobacteria. And when they check what are the taxonomies that are upregulated in this cis UCA and UVP, they are able to see that around 54 of these taxonomies are upregulated, out of which 9 are common in cis UCA and UVP. And among the ASV that are down-regulated, they are able to see that in cis UCA there are two ASVs. This is amplicon sequence that is down-regulated, and in the UVP they have seen that 13 amplicon sequence are down-regulated. However, there is no common sequence in between cis UC and UVP treatment. And when they check the phylum label results of 24 hours and compare it with the baseline, they are able to see the restorations of or the decrease in the firmicutes compared to the 8-hour results, and also they have seen some increase in this bacteroidetes for this mice that are undergone the cis UC levels. And for the mice that is exposed to UVP, they have seen a further they have seen an increase in these bacteroidetes, and uh, a decrease in the firmicutes. So this indicates that there is a different kind of resident bacterial species changes occurs when they are either exposed to CCUCA or UVP. And in terms of the common ASVs that are upregulated in CCUCA and UVP after 24 hours compared to the baseline is just 3, whereas around 18 CCUCAs are upregulated uh, compared to the baseline, and 47 ASVs are upregulated compared to the baseline. And when it comes to the ASVs that are down-regulated, around 31 cis UCA's levels are down-regulated, whereas for the UVP exposed mice, around 47 ASVs are down-regulated, out of which 6 are common. So this phylum levels further confirms the previous finding that it will change the resident bacterial species, and whether those change is helpful for the immune response by the skin, we will discuss those things in our subsequent videos. For today's videos, we have confirmed these changes in the resident skin bacterial species using the phylum label. In the previous video, we have also checked this resident skin bacterial species using different approach. So this this phenomena is confirmed by two different approaches. So this phenomena seems to be real rather than a artifact of the experiments. If you need to understand more about their experiment and their experimental results in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.